Good day everyone! Welcome to General Biology 1. Today, we are going to discuss about the different cell types and cell modifications. Now, if we can recall our previous modules, we learned the different characteristics of life and the different cell organelles and their functions that made the cell survive or that perform different processes in order for the cell to live. Now, for today, we are going to widen our knowledge on the different cell types other than the plant cell and the animal cell, as well as the cell modifications that made the cell adapt to the different environment in order for it to survive. So in here, we have our core concepts that will help us or that will guide us throughout our discussion. So the concepts that we are going to discuss are the following. First, we have the prokaryotic and eukaryotic cells. Second, we have the cell types of plant and animal tissues and their functions. Third, we have fungi, protists, and bacterial cells. And lastly, cell modifications. Okay, so first, the cell is the basic unit that contains the fundamental molecules of life. And we all know that the different characteristics of life are present or are manifested in the cell. That is why it is considered to be the basic unit of life. And of which all living things are composed. Now, all living organisms are made up of cells, as stated in our cell theory in your previous modules. A single cell is often a complete organism in itself, such as a bacterium or yeast. Now, a single cell organism, we can call it as a unicellular organism. Now, other cells acquire specialized functions as they mature. And when they mature, they work together with other specialized cells, forming now a more complex thing or more complex entity. And these entity, we can call it as tissues, and those tissues combine again to form organs and so on, which makes us, or which builds up now the multicellular organisms like us human beings and other multicellular organisms like animals and plants. So in here, we are going to differentiate the prokaryotic and eukaryotic cells. So first we have here the prokaryotic cells. Prokaryotes are the simpler and older of the two major types of cells. And prokaryotes are single-celled organisms. As you can see in the photo, in here, we have a single-celled organism and they are under the prokaryotic type of cells. Why are they called prokaryotic type of cells? Or why do they have this kind of cells? Because when you say prokaryotic cells, they do not have the major feature of a prokaryotic cell is that they do not have a membrane-bound nucleus embedded inside its own cell. So technically, the prokaryotes do not have a true nucleus, where all of their genetic information are stored or are located. So instead of having a nucleus, they have a free floating genetic material, okay? And that genetic material is located almost in the middle or in the central part of that cell, of that prokaryotic cell, and we call them nucleoids. So instead of having their true nucleus, they have nucleoids containing now their genetic material. So prokaryotic cells, they are mostly single-celled or unicellular organisms, and they do not have true nucleus in which their genetic material are enclosed or encapsulated. Now, for the eukaryotic cells, when you say eukaryotic cells, they have true nucleus. And as stated here, eukaryotic cells are thought to be the most modern type of cell. So from the prokaryotic cell, which doesn't have a nucleus, okay, it evolved into a eukaryotic cell, okay, seeing now a central structure containing the different genetic material of that cell, which is that structure is now called a nucleus. So all multicellular organisms, including you, your cat, and your plants or household plants, are eukaryotes because they all have true nucleus. Okay? So the big distinguished characteristic of these two types of cells is that prokaryotic cells do not have true nucleus, while eukaryotic cells have true nucleus. Other than that, prokaryotic cells are simpler in terms of their physical structure, in terms of their organelles, and most of them are unicellular organisms, while eukaryotic cells are a bit complex and have evolved from the prokaryotic organisms or prokaryotic cells, and they have a more complex structure and organelles. Okay, so that is for the differences of prokaryotic cells and eukaryotic cells. So in here we have an animal cell which is under eukaryotic cells. Okay, so animal cells have many different structures depending on their function. And we have discussed the different animals and plant cell organelles, okay? And they have their own function, and each function contribute to one another, and they are all interconnected in order for the cell to live and perform a lot of life processes. Okay? Next one, we also have here the plant cells. So plant cells have cytoplasm, cell membrane, and nucleus, which all perform the same function as animal cells. And if we can recall, the three fundamental parts of a cell is the cell membrane, we also have the cytoplasm, and the nucleus. The cell membrane is the outer covering or the barrier that is semi-permeable, okay? And then the cytoplasm is a fluid-like structure located inside the cell where all of the different organelles are embedded. And then the nucleus is the central structure of the cell where it contains all of the cell's genetic structure, and the nucleus is also the command center of the cell. Okay, so plant cell and animal cell have cytoplasm, cell membrane, and nucleus, and has same functions with the animal cells. However, there are organelles that are only present in plants and not found in the animal cells. Like for example, we have here cell wall, which is made up of cellulose. So cellulose is a complex sugar, okay, that are seen, or that are the main compositions of the plant cell wall. And this cell wall protects the cell membrane of the plants and the inner parts of the cell. Okay, next we also have the chloroplast, which is the site for photosynthesis. And if we can recall, okay, this type of, or this organelle is under the energy-related organelles, in which they are responsible, or they contribute to the development or creation of energy that is necessary for the performance of the different organelles of the cell, or of the cell as a whole. Okay, so the chloroplast contains the green pigment, which is the chlorophyll, and it traps or absorbs the sunlight energy, which is necessary for photosynthesis. That is why chloroplast is the site for photosynthesis. Next, we also have here the vacuole. Though animal cells do have vacuoles, but compared to our plant cells, animal cells have smaller vacuoles, while for the plant cell, they have a large central structure, or a large central vacuole that is used for water storage. Because as we all know, plants need water, or abundant or adequate amount of water in order for them to grow, and in order for their cells to be healthy. So this vacuole is too large that it almost occupy the whole cell. So these are the three organelles of the plant cell that is only present on this type of cell. Okay? Next, let's proceed. So other than the plant cell, we now have another type of cell, which is the fungal cell. Fungal cells are similar to plant and animal cells in that they have a nucleus, cell membrane, cytoplasm, and mitochondria. So meaning to say, fungal cells are under eukaryotic cells because of the presence of nucleus. And then, there are also the different cell organelles such as the cell membrane, the cytoplasm, and mitochondria because it is almost similar to the plant and animal cells. Now, like plants, fungal cells have a cell wall, but they are not made of cellulose. Instead, they are made up of chitin. So, for plant cells, they have a cell wall made up of cellulose, while for fungal cells, they have cell walls, but it is made up of chitin. So another complex substance that makes up the cell wall of fungal cell. An example of a fungal cell organism, or an organism that has fungal cell, or the fungi, okay? We have the different kinds of fungi. An example of that is a mushroom. So, a mushroom is a multicellular organism that has fungal cells, which also have a lot of different cell organelles that is necessary for the existence and survival of this mushroom. Okay, so that is for the fungal cell. Okay, let's proceed with the next cell type. We have here the Protist cell. So as stated here, Protists are eukaryotes, which means their cells have a nucleus and other membrane-bound organelles. Now, most but not all Protists are single-celled organisms because there are also some Protists that are multicellular organism. Now, other than these features, they have very little in common. So you can think about Protists as eukaryotic organisms that are neither animals, nor plants, nor fungi. Okay, so these types of cells do not satisfy the characteristics of an animal, a plant, or a fungi cell. That is why, okay, they are under this type of cells because they may have few characteristics of those other cells like the plant, animal, and fungi cells, but okay, not all the characteristics are present in them. So the scientists created this category of cells to place the different organisms, eukaryotic organisms, that do not fit in to the other types of cells. So we have here the three main points or main ideas that will help us understand more of the Protist cells. So number one, Ernst Haeckel set up the kingdom Protista in 1866. So it was Haeckel who created this category to place the different organisms that have very little or few common characteristics with one another, and they have few characteristics, or they do not satisfy the complete characteristics of the other cell types, the plant, animal, and fungi cells. Okay? So these unique organisms can be so different from each other that sometimes Protista is called the junk drawer kingdom because they cannot be categorized. There are very little characteristics or features. Okay? So that is why they are sometimes called junk drawer kingdom. So if a certain organism, okay, may have the characteristics of a plant cell, okay, they are, for example, photosynthetic, okay, they have cell wall. However, there are characteristics or features of that organism that do not satisfy the complete criteria for it to be categorized as part of the plant cell. So instead of what do you call this one? Instead of insisting that this organism is under a plant category, they Ernst Haeckel devised to have a new category, which is the kingdom Protista, placing now here the organisms that do not fit to the different cell types, to the other different cell types. Next, number 2, they are eukaryotic, which means they have nucleus. So protists have a nucleus in their cells, while some are photosynthetic, like what I have said earlier. Some are predatory, which means that they eat other organisms, or they eat other protists, and uses energy released through their mitochondria. So eukaryotic cells, or they are eukaryotic organisms, and some are photosynthetic, some are predatory, some are chemosynthetic. And for number 3, they can be parasites. Now, protists prefer aquatic and moist environments. That's their normal environment. Okay? Now, when these protists are introduced to another environment, like for example, ingested in the human body, okay? Such a protist may release substances, okay, in their body in order for them to adapt and to survive, okay, in that environment, because that's new for them. So, for them, the secretions were beneficial for their survival. However, these substances may be toxic to the host, or to, for example, the human body, because they are foreign materials for the human body, which is not normal in their or in our system. Okay, so that is for the different characteristics or common characteristics of protists. But other than these 3, okay, they have different features that make them distinct with one another. So let's proceed with the types of protists. So we have here 3 types of protists. We have the protozoa, the molds, and the algae. First, let's discuss the protozoa. So protozoans are unicellular organisms. And historically, they were called animal protists because they are heterotrophic. When you say heterotrophic, they depend to other microorganisms for them to live. So they make other microorganisms as food in order for them to get their energy. Okay? And they show animal-like behaviors, like they are predatory in nature. Okay? So protozoans are animal protists, okay, in the historical times, for being heterotrophic and show animal-like behaviors. They are also parasitic. That they are also parasitic protozoans that live in the cells of larger organisms. Like for example, when these protozoans, okay, since they live in aquatic and moist environment, like for example, in a pond, and when a certain organism is drank the water in the pond containing these protists, and they are ingested in that organism, okay, they become parasitic, okay, and they may impose diseases, okay, or even death to that organism in which they are ingested. Okay, so that is for protozoans. Now, protozoans, they do not have a definite shape. Okay, if you're going to look at your module, we have presented you the different examples of protozoans. So they do not have a definite shape. Okay, some protozoans have a disc-like shape. Some protozoans are oval in shape. Okay, some protozoans are shaped like a slipper, like for example the paramecium. Okay, while some protozoans, they do not really have a definite shape. For example, the amoeba. Okay, so the amoeba do not have a definite shape. Oh, so that is an example for the feature that makes them distinct with one another. And also, there are protozoans that are moving through the use of their flagella, okay? And there are protozoans that use cilia for them to move. Now, there are also protozoans that are both heterotrophic and also photosynthetic. Like for example, we have the Euglena. Okay, so the Euglena is categorized under a protozoa type of Protist because it is heterotrophic. But when the condition, when the condition is that there is abundance of sunlight, okay, in which since the Euglena, they have their own ways to develop their food through photosynthesis, so they do not hunt for other microscopic organisms for them to have their own food, okay, or for them to become their food. So they create their own food through the photosynthesis that they do, okay. So through the photosynthetic process, because they have similar organelles like the chloroplast in plants, okay, so in that way they create their own food. So in that way, the Euglena, being a protozoa, it has a different characteristic again. So the common characteristics of protozoans is that they are heterotrophic and they are unicellular. Other than that, okay, a definite shape, they do not have, not all protozoans have definite shape, not all have their similar ways to move, like some are ciliated or some have cilia, or while the others have flagella to move. And then another characteristic that differentiates the other protozoans to the other protozoans is the Euglena having that capability of doing photosynthesis. Okay, so that is for protozoa. Next, we have here the molds. So molds are saprophytic organisms, meaning they feed on the dead and decaying matter. So I think we are familiar with molds, like for example, the decaying bread, we can see molds there. Okay, so these type of microscopic organisms are under Protists. So they are divided into two types, which are slime molds and water molds. Okay, so they are tiny organisms that have many nuclei. So they have nuclei, they have many nucleus inside their cells. Okay, and the common characteristics of molds is that they are saprophytic organisms, or they feed on decaying matter. Okay, and then lastly we have algae. Now, these are generally single-celled or multicellular organisms. Okay, so these are photosynthetic, found mostly in freshwater sources or marine lakes. They are characterized by a stiff cell wall. So algae, they are single-celled. Okay, like for example, the blue-green algae, if you are familiar with that. Okay, also the brown algae, they are single-celled organisms, and there are also multicellular organisms. Okay, like for example, the red algae, okay, or the algae that is found in oceans. Okay, so they are photosynthetic in nature. They create their own food, and mostly found in marine areas. So these algae contribute also in the creation of oxygen that is necessary for us to live. Okay, because they do photosynthesis. Okay, so as you can see, if you're going to observe, okay, there are different characteristics from being a protozoa, from being molds, from being algae. They are different with one another. Some may satisfy, or some may have the characteristics of plants, like being photosynthetic. Some may have that characteristics of animals, being what do you call this one? Being heterotrophic, depending on, or feeding themselves with other microorganisms. And some are like fungi, because they eat decaying matter. Okay, but the other characteristics, they do not satisfy the other cell types. So that is why they are under the Protist cells. Okay, next let's proceed. So the protozoans can be divided into 4 major groups. So we have the amoeboid protozoans, first one. So mostly found in water bodies, either fresh or saline. They have pseudopodia, okay, a false feet, which help them to change their shape and capturing and engulfing food. So the example here is the amoeba. So this is under the protozoan category of the protists. Okay, so amoeboid protozoans. So their mode of movement is through the false feet or the pseudopodia. Okay, next, number 2, we have the flagellated protozoans. As the name suggests, meaning to say they have the flagella or the flagellum, okay, for them to move. So they are free living organisms, or they are moving. Okay, they float everywhere. They are everywhere. Okay, they are free living. They are also parasitic in nature. So an example of this one, flagellated protozoa, is the euglena. Okay, and we also have here ciliated protozoans. So ciliated meaning to say they have cilia all over their body for their locomotion. Okay, as well as their nutrition. So they are always aquatic. Okay, so an example of a ciliated protozoan, we have here the paramecium. So basing on these three, okay, with these three groups of protozoans, we can see immediately that they are distinct in the way they move. Okay, the pseudopodia in the amoeba, flagellum for the euglena, and cilia for the paramecium. However, they are still in that protozoa group. Okay, because they are heterotrophic in nature. They engulf food. They catch prey. They eat other microscopic organisms, and they are parasites that mostly live in fresh or saline water. Okay, another group of a protozoan, okay, is the sporozoans. So these organisms are so called sporozoans because their life cycle has a spore-like stage. So in their life cycle, there is a spore-like stage, okay, as they develop. So an example of this is the malarial parasite, which is the Plasmodium. Okay, so they are all parasites. They have their own characteristics, and they have commonalities with one another. That is why they are under protozoa. Okay, so that is for the three types of protists and the 4 major groups of protozoans. Okay, next let's proceed. So let's proceed with the cell modifications. So cell modifications are specialized modifications reacquired by the cell after cell division that helps the cell in different beneficial ways. So they do modifications in order for them to adapt, okay, to adapt to the new environment, to adapt for them to thrive and survive that environment. So those are the modifications that were made by cells, okay, for their survival. So first, there are 3 types of cell modifications and examples of each type that have their function. So first, we have the apical cell modification. As is stated here, found on apical or apex or tip surface or surfaces of the cell. Okay, next we have here the basal cell modification. So if the apical cell is on the apex, the basal cell, okay, the modification that is made on the base surface of the cell. And then we also have the lateral cell modification, okay, where the cell modification is found on the lateral surface of the cell. So we are going to discuss them one by one. First, we have the apical cell modifications. So the apical cell modifications are found on the tip of the surface of the cell. So we have here, okay, we have the cilia and flagella. So, as we all know, cilia are short hair-like structures that move like waves. Okay, so in this example, in this picture, we have here the paramecium, which is a ciliated microorganism that is surrounded by cilia, and this cilia is used by the paramecium for them to move from one place to another. Okay, and these cilia, they do the wave-like movement for the paramecium to transport itself or to move. Okay, another modification that was made for locomotion is the flagella or the flagellum. So long whip-like structures that are formed from microtubules. So as you can see here in this structure, they are like tails of that microorganism. Okay, so we have here the flagella with the same function with the cilia. However, the modification is that there are only two flagellum, okay, two elongated whip-like structures. Okay, but still they are used for locomotion. Okay, so the movement now for flagella is like a propeller. Okay, propeller-like movement. While in cilia, it is wave-like movement. So that is the modification for locomotion. Now let's proceed with the other apical cell modifications. We have here the microvilli. So the microvilli is a finger-like projections that arise from the epithelial layer in some organs. They help to increase surface area for faster and more efficient absorption. So the microvilli is used for the absorption of nutrients that are digested by the different organisms. Like for example, in our human body, we have this kind of finger-like projections, and it is found in our intestines, specifically in our small intestine. Okay, so in our small intestine, there are finger-like structures, the villi, and then within or in that surface of the villi, there are even a smaller finger-like projections. That is now the microvilli, which is for the absorption of nutrients that was digested, okay, or that was grind or broken down into smaller pieces in the stomach, and now the intestines, okay, the small intestine, through the help of this microvilli, it absorbs the nutrients that is necessary for the cells. Okay? Next modification, we have here the pseudopods. Okay? So, as the definition say, temporary irregular lobes formed by amoebas and some other eukaryotic cells. It bulges outward to move the cell or engulf prey. Okay, so these pseudopods, okay, they are false feet for the amoeba to move. Okay, so it seems like they are jelly-like structures in which they project out. Okay, they bulge. Okay, they bulge from the main cell. Okay, and that bulge will pull the cell into that direction, making that unicellular organism move. Okay, other than that, when there is a nutrient or other microscopic organism that can be their source of food, okay, the pseudopod will attach themselves or will go to that direction and engulf, okay, that microscopic organism or prey or that nutrient. Now, another apical cell modification we have here, the extracellular matrix. So, the compound secreted by the cell on its apical surface. For example, the cell wall is the extracellular structure in the plant cell. Same is true with the other cells like the fungi cell, because they have their own cell wall made up of chitin, while for plants, they are made up of cellulose. Okay? And glycoprotein is the main ingredient of extracellular matrix in animal cells. So as you can see here, this is an example of animal cells where they have their own extracellular matrix. They are mainly composed of glycoproteins and other microscopic materials, like for example the proteoglycan molecule, we have the polysaccharide molecule, and the collagen fiber. These are the parts of the glycoprotein that surrounds, okay, that is the component of the extracellular matrix of the animal cells. Okay, so that is for the apical cell modifications. Now let's proceed with the basal cell modifications. Now these are cell modifications that are made on the base surface of the cell. So we have one, the hemidesmosomes. So hemidesmosomes are anchoring junction on the basal surface of the cell. So they are rivet-like links between cytoskeleton and the extracellular matrix components, such as the basal lamina that underlie epithelia. So as you can see here, okay, they are anchoring junctions. So they attach, okay, they are attachments to the base of cellular structures. So these hemidesmosomes, okay, they are they have the function of anchorage, okay, that attach themselves to the base surface of the cell. Okay, so the cytoskeleton and extracellular matrix components, okay, they are attached to the basal lamina because of the help of the hemidesmosomes. Okay, next we have the another type of cell modification. We have the lateral cell modifications. Now, these type of modifications are made on the lateral surfaces of the cell. So, in our picture here, okay, we can see three junctions. Okay, these are the lateral modifications. Okay, first we have the tight junction. They act as barriers that regulate the movement of water and solutes between epithelial layers and prevent leakage of extracellular fluid. Okay, so they are the barriers. Okay, so these tight junctions, since they are tight, they block, okay, or they prevent the moving out of different solutes and water molecules inside the or that are found in the extracellular fluid or part of the extracellular fluid. Okay, so that is for the tight junction. Now we also have here the adhering junctions. Okay, they function in fastening cells to one another. So they fasten. Okay, they join cells with one another. Okay, and then last we have the gap junction. Okay, they are the communicating junctions that connect the cytoplasm of adjoining animal cells. Okay, so they serve in the communication between cells. Okay, next, so those modifications, okay, they are made for the cells to adapt, okay, and for their survival. So for adaptation and survival. Now, specialized cells are cells that are structured to fit a certain function. These cells are modified, unlike the rest that is reproduced into the standard. So to what cause? To what cause that these modifications are made? So to serve a purpose only they can achieve because of their specialized structure. Okay? So these modifications, they may be used or they may be modified, okay, but their purpose, okay, they have their own purpose which contribute to the existence of the cell and for the different processes to be executed well, like the communication of cells with one another and other processes. Okay, so I hope you learned something with this simple lecture video that we have made. So read more on your modules, because the module gives a detailed discussion or gives a lot of pieces of information, okay, that you can use as reference. Okay, so the module is a guide for you. Thank you very much and God bless.